Welcome to JO&SAM Healthcare Technology. Today we are going to see about a diagnostic equipment spirometer. We all know that the function of lungs is to take in oxygen and eliminate carbon dioxide. Spirometer is an instrument to assess the pulmonary function and to identify the pulmonary impairment and then to quantify it. Now, let us start with the basics before we dive into the spirometer working. At first, interchange of gas between an organism and the medium is called as respiration. There are two types of respiration. One is the internal respiration and the other is the external respiration. Internal respiration is nothing but the exchange of gases between the bloodstream and nearby cells. That is the respiration that takes place in our tissues. Now, external respiration is the exchange of gases between the lungs and the external environment. That is the gas exchange that occurs in the lungs. The representation of the same has been shown here. The deoxygenated blood from the tissues goes to the heart, and from the heart it goes to the lungs for purification, that is for becoming oxygenated. So in the lungs the gas exchange takes place. Now the oxygenated blood from the lungs reaches the heart, and the oxygenated blood from the heart is pumped through the tissues. It is important to note that the oxygen and carbon dioxide ratios are different. The blood leaving the tissues and entering the heart has more carbon dioxide than the oxygen level. Now, if we compare the carbon dioxide and oxygen of the blood that goes from the lungs to the heart and then to the tissues, what happens is the oxygen level is much more greater than the carbon dioxide. Next is external respiration. When we speak about lung function, we have to concentrate on external respiration, right? Okay. Now, so what is external respiration? As we saw earlier, it is the gas exchange occurring in the lungs. In order to respire, the organs of the respiratory system works together. That is, it acquires the oxygen and eliminates the carbon dioxide. The respiratory organs are nostril, nasal cavity, pharynx, larynx, trachea, bronchus, bronchioles, and alveoli. When we respire, there may be microorganisms in the atmospheres. So airborne particles, toxic substances, microorganisms, etc. has to be eliminated from entering into the lungs during respiration by these respiratory organs. The organs of respiration are divided into two divisions. That is, one is the conducting division and the other is the respiratory division. When we speak about conducting division, it is the place where there is no gas exchange. And in respiratory division, the actual gas exchange takes place. So what are the organs that the conducting division has is the nostril, nasal cavity, pharynx, larynx, trachea, bronchus and some parts of bronchioles. When we speak about respiratory division, it is the place where actually gas exchange takes place, right? So the organs of the respiratory divisions are respiratory bronchus, alveolar duct and alveoli. We have seen the organs of respiration till now. Next, I am going to highlight important points about the respiration process. That is, the process of respiration involves inspiration and expiration. Inspiration is nothing but breathing in, that is, taking the air in, whereas expiration is breathing out, leaving the air out. The respiration cycle consists of both the inspiration and expiration. So the number of respirations or breaths per minute is called as respiration rate. It differs for both adults and children. So adults have a normal respiration rate of 12 to 20 breaths per minute, that is BPM. But for children it may be as high as 40. Next, we are going to see the mechanics involved in breathing. When we say about mechanics, there are two sets of muscles that are majorly involved in breathing. One is the diaphragm and the other is the intercostal muscle. So when we see about diaphragm, it is the wall that separates the abdomen from the thoracic cavity. And also the diaphragm is a dome shaped structure, that is it will be looking like this, and then it is responsible for moving the thoracic cavity up and down. That is, during respiration what happens is this diaphragm is responsible for moving the chest up and down. And next is the intercostal muscle. So intercostal muscle is the muscle that separates, intercostal muscle is the muscle that surrounds the thoracic cavity and is responsible for moving the chest in and out. Next, we are going to focus how these two muscles are involved in the process of respiration. That is, during inspiration, what is happening, and during expiration, what is happening. So during inspiration, both the diaphragm and the intercostal muscles are contracting. The diaphragm exhibits a downward movement and the intercostal muscles moves upward and outward. And during expiration, what happens is both the diaphragm and the intercostal muscles relaxes. So the diaphragm exhibits an upward movement and intercostal muscles exhibits an inward and downward movement. So in this diagram, as you can see, what happens during inspiration and expiration. So what happens to the diaphragm? So diaphragm moves downward during the inspiration and upward during the expiration. And what happens to the intercostal muscles? Intercostal muscles moves upward and outward during the inspiration and downward and inward during the expiration. So if you sit normally in a place and then you relax and you breathe, so that you can feel the inspiration and expiration. Okay, till now we have discussed about respiration, types of respiration, external respiration, mechanics of breathing, etc. Now we are going to see what are the measurements that can be made on the pulmonary functions. There are three types of measurements that can be made, that is ventilation, distribution and diffusion. So ventilation is used to determine the ability of the lungs to move the air volumes and the speed at which the air is moving. Okay, so next is the distribution. Distribution measurements provides information on the air flow, majorly on the air flow, that is how the air flows in the lungs, and it indicates if any disease is present or what. Next is the diffusion measurement. So diffusion measurement, before seeing what is diffusion measurement, we all know that in the lungs the oxygen and carbon dioxide is exchanged only due to the process of diffusion, right? So oxygen is taken and carbon dioxide is given out by the process of diffusion. So diffusion measurement represents the exchange of gases, that is the diffusion of O2 and CO2. Let us concentrate only on ventilation in order to see about a spirometer. Okay, there are certain parameters that can be measured related to lung volumes and capacities that we are going to see. So first is the lung volume. In that we are going to see the volumes such as tidal volume, inspiratory reserve volume, expiratory reserve volume and residual volume. When we see this diagram, so the upward curve represents inspiration and downward curve represents expiration. The tidal volume of air is the amount of air that can be either inspired or expired during each cycle. So this tidal volume is normally 500 ml for an adult. Next is the inspiratory reserve volume. So inspiratory reserve volume is the additional amount of air that can be inspired after normal inspiration, right? So that is, uh, as you can see, which is a normal inspiratory end here. So this is the normal inspiratory end here. Okay, so and after that how much you inspire forcefully, this is your inspiratory reserve volume. So this is your inspiratory end, and after your inspiratory end how much air you can forcefully inspire is your inspiratory reserve volume. And next is the expiratory reserve volume. So expiratory reserve volume is the additional volume that can be expired after a normal expiration. So that is nothing but this is your expiratory end, that is the normal expiratory end, and after which you can expire forcefully is your ERV, that is from this end you consider. So from this end if you expire forcefully, that becomes your ERV. And even though if you forcefully expire, there will be for sure some amount of air that will be remaining in the lungs. So that is what your residual volume is. So that is being represented in orange color here. So even after if you forcefully expire, there will be some amount of air remaining in the lungs. That is called as your residual volume. Next, we are going to see about the lung capacities. So the lung capacities are inspiratory capacity, functional residual capacity, vital capacity and total capacity. So the first we are going to see about inspiratory capacity, the maximum volume inspired from the resting and expiratory position. So which is your resting and expiratory position here? So this is your respiratory end, that is your especially your expiratory end position, right? So from this you can how much volume of air you can inspire, that is this TV plus your IRV. So this is the amount of air that you can uh, inspire max at a maximum. So that is what your inspiratory capacity is. And next is your functional residual capacity, that is your volume remaining in the lungs after normal expiration. So which is your normal expiration here? So this is your normal expiratory end, and from here what is the amount of air that is remaining in the lungs? So your ERV plus RV becomes your FRC, that is your functional residual capacity. Next, vital capacity. Vital capacity is the maximum volume of gas inspired voluntarily after maximum expiration. So which is your maximum expiratory end here? So this is your maximum expiratory, that is your forceful expiratory end. And after this, how much air you inspire? So from this end you can inspire ERV, then TV, then IRV. So all these three together forms your vital capacity. Now the last capacity is the total capacity, that is nothing but the volume in the lungs at the point of maximal inspiration. So if you have maximal inspiratory point, this is your maximum inspiratory point. And what is the amount of air remaining in your lungs now? So IRV plus TV plus ERV plus RV becomes your total capacity. So the combinations can be different. As you can see, IC can be given by the summation of IRV plus TV. FRC can be given by the summation of ERV and RV, and VC can be given by the summation of IC plus ERV. Then TC can be given as a summation of IC plus FRC, or VC plus RV, or the summation of IRV plus TV plus ERV plus RV. So the combinations can be different. Next, there are certain other things that you can consider when you speak about pulmonary function. That is your airway resistance, your lung compliance, lung elasticity. Airway resistance tells us how easily air flows within the lungs. Say suppose if the airway resistance is high, then what happens is the respiratory structures and lungs will have a reduced airflow. Next is the lung compliance. Lung compliance is the ability of the lungs and tissues to expand on inspiration. If there is no proper expansion of the tissues, what happens? The inspiration gets affected. Okay. Next is the lung elasticity. During expiration, our lungs should recoil to normal condition. So elasticity is the ability of the lungs to recoil during expiration. Now finally, we are going to see about the spirometer. So what is spirometer? A spirometer is an instrument used to measure the lung capacity and lung volumes. So we have discussed about lung capacities and volumes in the earlier slides. So the spirometer is an instrument that measures the lung capacities and the volumes. The the spirometer shown here is a conventional one, which is a water seal spirometer. The nose of the patient is clipped, as you can see here, in order to keep the nose closed, and the patient respires only through the mouth. Here a water filled cylinder is present, and an inverted floating drum is suspended on the water. Now this floating drum is connected to the counterweight. Here is the counterweight. So this floating drum is connected to a counterweight balance with a string through this pulley. Okay, this is a pulley over here. And also here a pen is attached to have a record of the chart, which is a rotatory moving drum called the kymograph. As we inspire and expire, the volume inside the floating drum changes, and this change is translated into motion where we are able to record it. Now we are going to see what happens during inspiration and expiration process. Say suppose the patient inspires. What happens? The floating drum drops. That is the pressure inside the floating drum drops, and hence the floating drum moves downward. So if we notice the counterweight that is attached through a pulley with this floating drum, it moves upward. So pen also moves out upward. As a result, we get an upward curve in the graph during inspiration. So if we observe what happens during expiration, it is just opposite to what happens during inspiration. The pressure inside the floating drum rises, so it moves upward. The counterweight and pen moves downward. Now we get a downward curve during expiration. When weight is attached to a potentiometer, electrical signal is produced as output. The output produced by the pen and electrical signal are almost similar results. So nowadays we don't have such kind of spirometers. We have electronic versions of spirometers. So there a sensor or transducers can be used where you flow, where the air flows from your mouth. That is, you blow on the sensors. So the sensor may be a different thing. That is, say a turbine sensor, then pressure differential flow sensors, ultrasonic sensors can be used. Okay, we have finally arrived to our conclusion part. So, in obstructive, there are two types of lung diseases. That is, one is obstructive and destructive. In the obstructive lung disease, there will be problems during expiration. As you can see here, there may be some obstructions in your airway. So, what happens is the airway resistance will become high. In restrictive disease, people will have problems in inspiration. Lung compliance will be low. That is, the lung is struggling to expand. So that is what your low lung compliance indicates. We can arrive at certain parameters to find obstructive or restrictive lung disease is present. First one is the forced vital capacity. It is the amount of air that can be forcefully exhaled from the lungs after taking a deepest breath possible. That is, when forcefully we inspire and then forcefully we expire, we get the FVC. And the second one is the forced expiratory volume. So this is the maximum amount of air that can be forcefully exhaled in 1 second. And the ratio of these two, that is the forced vital capacity and forced expiratory volume, becomes another parameter. So I have listed the few measurements to indicate whether if it is low or high, to indicate whether it is a obstructive or restrictive disease. So FEV1, FEV, the ratio of FEV and FVC, residual volume, total capacity, and the vital capacities are provided here. Say suppose if we take the ratio of FEV1 by FVC, if suppose it is less than 70 percentage, it indicates airway obstruction. If the ratio is greater than 85 percentage, then it may be a restrictive disease. So even with other parameters, it is possible to find whether a person has obstructive or restrictive disease. For example, we know asthma, right? So asthma is a obstructive disease where we have problems during expiration phase. And uh, say suppose a person is having pulmonary fibrosis, that is a kind of restrictive disease. With this, I would like to end the video. So we have discussed about the respiration, types of respiration, then what is external respiration, the mechanics of breathing, lung volumes and lung capacities, and we have seen what is the spirometer, and we have seen about different kinds of diseases, that is the lung diseases, obstructive, restrictive diseases, and we have seen how the parameters can be used to detect the obstructive and restrictive diseases. The references are R.S. Kanpur, Handbook of Biomedical Instrumentation, Elaine N. Marriott, Katja Hoyen, Human Anatomy and Physiology. Thank you.